CD8 is a transmembrane glycoprotein that serves as a co-receptor for the T-cell receptor. Like the TCR, CD8 binds to a major histocompatibility complex molecule, but is specific for the class I MHC protein. There are two isoforms of the protein, alpha and beta, each encoded by a different gene. In humans, both genes are located on chromosome 2 in position 2p12. Tissue distribution. The CD8 co-receptor is predominantly expressed on the surface of cytotoxic T cells, but can also be found on natural killer cells, cortical thymocytes, and dendritic cells. It is expressed in T-cell lymphoblastic lymphoma and hypopigmented mycosis fungoids, but is frequently lost in other T-cell neoplasms. Structure. To function, CD8 forms a dimer, consisting of a pair of CD8 chains. The most common form of CD8 is composed of a CD8I plus or minus and CD8I squared chain, both members of the immunoglobulins superfamily with an immunoglobulin variable-like extracellular domain connected to the membrane by a thin stalk, and an intracellular tail. Less common homodimers of the CD8I plus or minus chain are also expressed on some cells. The molecular weight of each CD8 chain is about 34 kDa. The structure of the CD8 molecule was determined by Lee, D.J., Axel, R., and Hendrickson, W.A. by X-ray diffraction at a 2.6A resolution. The structure was determined to have an immunoglobulin-like beta sandwich folding and 114 amino acid residues. 2% of the protein is wound into I plus or minus helices and 46% into I squared sheets, with the remaining 52% of the molecules remaining in the loop portions. Function. The extracellular IgV-like domain of CD8 I plus or minus interacts with the I plus or minus 3 portion of the class I MHC molecule. This affinity keeps the T-cell receptor of the cytotoxic T-cell and the target cell bound closely together during antigen-specific activation. Cytotoxic T-cells with CD8 surface protein are called CD8 plus T-cells. The main recognition site is a flexible loop at the I plus or minus 3 domain of an MHC molecule. This was discovered by doing mutational analyses. The flexible I plus or minus 3 domain is located between residues 223 and 229 in the genome. References. External links: T-cell group, Cardiff University, mouse CD antigen chart, human CD antigen chart.